Hello, everyone. Thank you very much for joining this afternoon. We will just give another minute for some more people to join, and then we will go ahead and get started. All right, so we will have some time for some more folks to filter in, but I will go ahead and begin with introductions. So usually we would have a set of slides to go along, but unfortunately we're having some technical difficulties, so we will proceed without the slides. But we wanted to welcome and good afternoon to everyone today. Thank you so much for joining our webinar, which is hosted by our program Liver Cancer Connect. My name is Beatrice Sovich, and I'm a public health program manager at the Hepatitis B Foundation and the manager of our Liver Cancer Connect program. So thank you all for tuning in today. Today's webinar is going to be focused on the work and research of Dr. Anand Mehta, who has done extensive work on innovative methods for the early detection of hepatocellular carcinoma or primary liver cancer. It has become increasingly clear in recent years and decades that HCC is too often detected when it is at an advanced stage, at which time treatment is much more difficult, and overall survival rates are much lower and greatly decreased. So in honor of Cancer Awareness Month this February, we'll have an opportunity today to hear from Dr. Mehta, who will share an overview of the findings from his recent paper, highlighting the development and application of a new biomarker platform that can contribute to earlier detection of hepatocellular carcinoma while the disease is still in its earlier stages and is more treatable and less deadly. So we are very much looking forward to this great presentation today. Just a quick note about participating in Zoom webinars before we get started. I would just like to remind all participants that during the webinar, all audience members are muted, but we encourage you to use the chat feature to let us know who you are and where you're joining from. And if you have any technical challenges, please indicate your issue using the chat box, and I will help to address it as soon as possible. And if at any time during the presentation you have a question, please feel free to enter it into the Q&A box. Questions will be addressed at the conclusion of the presentation, and the session is also being recorded. We'll share out the recording with all webinar registrants at the conclusion of the event. And then just to provide a brief overview of our Liver Cancer Connect program. So Liver Cancer Connect is a dedicated program of the Hepatitis B Foundation, and it was established for the explicit purpose of raising awareness about liver cancer, both that arises from hepatitis B, which is its most common etiology, as well as from hepatitis C, MASLD, and other etiologies as well. And also of providing support to those living with and affected by liver cancer through a variety of channels, including email and phone consultations, social media, blog posts, a website, and webinars and other events such as this one. So we also run a liver cancer focused events and lead liver cancer public health research projects as well, including most recently our Learn the Link campaign, which used the findings from various focus groups that were conducted within communities heavily impacted by hepatitis B to inform a culturally and linguistically relevant communication campaign, highlighting the connection between hepatitis B and liver cancer and encouraging screening tailored to each different community. So we have 12 communities total, and we are hoping to continue to build and expand our programming throughout 2025 and the years to come, and we'll keep you updated about these developments and further opportunities to get involved. So now to move on to introduce our speaker today. So we're very excited and honored to be joined today by Dr. Anand Mehta, who is the Smart State Endowed Chair and Proteomic Biomarker Professor at the Medical University of South Carolina. Dr. Mehta is interested in understanding and developing diagnostic methods and treatments for primary liver cancer or hepatocellular carcinoma. And he has developed a method referred to as glycoproteomics and has used this method to identify several serum biomarkers of liver disease and liver cancer. His lab was one of the first to perform serum glycoproteomics, And through this, Dr. Mehta's group has identified over 30 serum glycoproteins with increased levels of glucose levels in those who are living with hepatocellular carcinoma. So some of these identified proteins are already being used clinically in China for the detection of HCC, and others are being used in the USA to detect fibrosis and cirrhosis and some of the precursors of HCC in those living with metabolic dysfunction associated steatotic liver disease, or MASLD. So we're very excited to hear from Dr. Mehta today, and we're grateful to him for joining us. We again invite audience members, if you have any questions along the way, please feel free to put them in the Q&A box, and they will be addressed at the conclusion of the webinar. So without further ado, I pass it on to Dr. Mehta to begin your presentation. Thank you so much again.
Thank you. If I can oh, perfect.
Yes,
I can
sorry, here we go.
Share now. And hopefully everybody can see that, and we will go.
Yep.
So, first of all, thank you again for allowing me to present my work. And as academics, we always like to talk about our work, so I appreciate the time and the participation of everybody here. So, I'm going to talk a little bit today about what we've been doing to sort of find biomarkers of liver cancer, but also I think address some of the areas which have caused us to fail in our search for biomarkers of liver cancer. And I think, we've learned a lot of things along the way, and I think we're getting to a point, I think, where we're now able to leverage new technologies to really, I think, finally get something that's useful. Sorry. So as many of you may know, there are really two major types of liver cancer. There is hepatocellular carcinoma, which is a tumor that arises primarily from transformed hepatocytes, and a cholangiocarcinoma that can arise from cholangiocytes that have become malignant. And those two represent primarily the two major forms of primary liver cancer that you'll see. There are combined types, there's rare types that occur in pediatric conditions, but for the most part, these are the two major types of primary liver cancer. And hepatocellular carcinoma represents the major form, and it's still, a shocking statement that almost, 35 years after the vaccine was developed, hepatitis B remains the major causative agent of liver cancer worldwide. There are still close to 900,000 cases of liver cancer worldwide. And even now with the cure of hepatitis C, it's still a major etiological agent of liver cancer. In the US, though, the major etiologies are really alcohol and a high-fat diet, now referred to as MASL, metabolic associated liver disease. And so what's coming, with the new drugs that treat, obesity, if that will change, we'll see. But today I'm really going to focus on the work we've done for hepatocellular carcinoma. But for all intents and purposes, everything I will say about hepatocellular carcinoma, we're actually also doing for cholangiocarcinoma, because some of the exact same methods apply, and we have projects going on to look at both of these. So the one thing about HCC that's very important is that, unlike many other cancers that we've gotten very good at treating, whether it's breast cancer or lung cancer or even colon cancer, there are very limited chemotherapeutic options for HCC. And so really, the best outcome is still obtained when the tumor is identified early, and then you can resect it or ablate it, but usually resect it. And that patient can then go on and live, for a good long period of time. This is actually our data, but it mimics what others have seen, so that if a tumor is caught early and surgically resected or transplanted, overall survival is generally greater than 60 months. It's actually very, very good. In contrast, if the tumor is caught at a later stage, where the tumor is either spread in multiple locations within the liver, or it's got to a point where it's very large within the liver, that makes surgical methods un unrealistic. Overall survival is actually quite poor in less than 20 months. And so for that reason, early detection is absolutely essential for this tumor type. And because of that, there are very defined screening guidelines or surveillance guidelines for those at risk. So for the most part, we know who's at risk of developing this type of cancer. That's people who have some degree of significant fibrosis, cirrhosis, either driven by alcohol or hepatitis C or fatty liver disease, or have chronic hepatitis B. And so in those patient populations, the recommendation is that every 6 months they get screened either by an ultrasound plus a biomarker, I mean, that biomarker is AFP, or by some imaging methodology to look for the presence of liver cancer. Because we know those patients are at the highest risk of developing liver cancer. And that can be up to an annual risk of 5% per year, which is quite significant. And so imaging modalities are expensive. They're, in some cases, not optimal. Things like ultrasound are not optimal in certain patient populations. And people have been looking for new biomarkers that can complement ultrasound or even replace ultrasound that can be then used to bring people into the more advanced imaging methodologies like MRI or CAT scan. And people have been trying for years, and we've been trying for years to find a biomarker that actually could be used in that situation. And I think we've learned a lot along the way. We've learned, I think we've learned to a certain extent why what we were trying to do has not really worked so well. And I'll show you some of that data as well. So one of the key goals of any biomarker is you want to have some association actually with the tumor itself. And so a lot of times, when we think of biomarkers, they are just something in the blood or in the saliva or in urine or in fecal matter, some fluid that is indicative of a disease or a process occurring in a person. And so, in the case of a tumor biomarker, you want to have that biomarker really be associated with the tumor. And so, for a lot of stuff that we're doing now, we're actually starting with direct tumor tissue and identifying what's going on within the tumor. And then we used matched serum to see, can we see what's happening in the tumor? Can we see it also in the serum? And can we kind of have some biological relevance to that fact? Um, and so, and I think it's true for any biomarker. You want to have some association with the tumor. And in this case, here's a piece of tissue, and there's a tumor within that. I am not a pathologist. I'm not an MD, and so I need the help of a pathologist to annotate this tumor and tell me where the actual tumor is. And a pathologist can do that, and many of you may actually be able to do this as well. But the pathologist can annotate the tumor, and that tumor area is within that circle that was annotated. And ideally, you would like something like this. This is just an analyte that we measured using a method I'll describe in a little bit. But it shows a color scale where blue shows, very little of that analyte, that molecule. And then green and red represents a high level of that molecule, or a higher level of that molecule. And as you can see, in this case, this analyte is highly associated with the tumor region and not with the surrounding region. So that would make, if we could figure out what that was, and then find that also in the bloodstream or in urine or fecal matter, whatever, that could be a good biomarker. That's the goal. And so, again, you would like something like that or something like this. So, again, you don't have to be a pathologist, and I won't pretend to be a pathologist, but if you had to guess where the tumor region was, you would probably do what I did and say that highlighted area that's showing this high level of this analyte is actually the tumor, and that's actually correct. And we've had pathologists annotate all this tumor. And so you want a biomarker that looks kind of like that or like that. And again, you don't have to be a pathologist, but I can guarantee you that's where the tumor is. And this analyte that we're measuring is showing up directly in the tumor, and like that, and like that, and like that, and like that, and like all of these samples. And so in all of these samples, what we're actually measuring are sugars on proteins. And these are called glycoproteins. And so a glycoprotein is nothing more than a protein with a sugar attached. That's all it is. And so people know proteins, but they don't realize that but most proteins have things attached to them. And glycoproteins are proteins that have sugars attached to them. And a protein often can have different sugars attached to them, and that's called a glycoform. So the term isoform is something we've often heard in terms of proteins, and a glycoform is just a protein with a different sugar attached to it. And that sugar can fundamentally change the function of that protein. So many people know immunoglobulin. So immunoglobulins are proteins that have a specific function to mediate an immune response. And that immune response, how that antibody will actually elicit an immune response or drive an immune response, is actually controlled entirely by the type of sugar attached to that protein. And so these things have functions. Now, what's also interesting is that many biomarkers are actually just sugars that are either attached to proteins or actually proteins. And so, for example, one of the most commonly used biomarkers is CA19-9. It's used for pancreatic cancer, for bile duct cancer, for colon cancer, and stomach cancer. That's actually just a sugar attached to lipids and proteins. And people don't even look at the proteins, they just look at the lipid. And they just look at the sugar. And so that's a protein, that's a biomarker that we know exists that's actually out there. CEA, which is a carcinogen antigen, an embryonic antigen. It's used in colon, prostate, ovarian cancer. That's just a sugar attached to protein. The mucins, MUC5A, MUC5AC, 51, MUC1, that are often used for lung cancer, pancreatic cancer, colon cancer, are actually just sugars attached to proteins. And in the case of liver cancer, we actually have one as well called AFPL3. That's a sugar version of AFP that's actually a better biomarker for liver cancer. And so there's already lots of evidence where sugars on proteins or on lipids have been used as biomarkers of cancer. And to be honest, they work okay. They don't work great, but they work okay. And I'll show you some of the data of why they only work okay in a little bit. Now, what we developed at MUSC is a method to do tissue analysis um, to look at analytes in tissue. And so all of our workflows at MUSC now involve looking at tissue that involves tumor tissue and then also having matching serum from those individuals. So we can look at the tumor tissue and we can look at serum from those same individuals to see, can we see what we see in the tumor itself in the bloodstream, and that can that be then leveraged as a biomarker? That's the goal, and so that's the kind of how we switched things around. 10 years ago, actually, it's about 20 years ago when we started this, we didn't actually have good ways looking at tumor tissue. We looked at really just serum, and we didn't know if what we were looking at came from the tumor or not. And so we always struggled with that. And so this method, I won't go into too much detail about it, but it generally uses very defined and well-known workflows with dealing with tissue. So anybody that's ever done any work with tissue can prep a slide using this method. It's actually very common. It's normal histology and pathology workflows. The difference is that instead of adding, for example, an antibody to stain a certain protein, what we do is we spray on a thin layer of enzyme onto that tissue that allows us to look at certain analytes. And so we can look, for the most part, at anything in that tissue. We can look at lipids, small molecules, metabolites, proteins, extracellular matrix proteins, and we can look at sugars that are attached to those proteins using a wide variety of proteins that we actually started to make in Doylestown. So I think almost 15 years ago. And then we then analyze that tissue in one of these high-end mass spectrometers. So that's the expensive part. And so these high-end mass spectrometers literally fire a laser across the tissue at every 10 microns and analyze and get huge amounts of data, basically thousands and thousands of data points across that tissue that allow you then to look at, what you're seeing in a spatially resolved way. And I'll show you what that looks like. So what it looks like is this. So let's go back to that one piece of tissue where, again, literally, if you imagine that tissue as being an XY coordinate, so literally, X here, Y here, and each XY coordinate is broken down into a small little area of, say, 10 microns. We've analyzed that by the mass spectrometer and we've collected data over the entire tissue. And so you can go back and tell the computer or ask the computer, hey, computer, like what, for example, was not present in this area, but more present in the surrounding area. and it'll spit out a sugar that looks like this and say, hey, this molecule that has this M over Z value is weakly found in the tumor area, but more abundant in the adjacent area. At the same time, you can then go back and say, hey, computer, what's found in the tumor area and not in the adjacent area? And it can spit out a sugar like this, that again is found in the tumor area and not in the adjacent area. And so, and that way we get an idea of what's happening in the tumor and what's happening in the adjacent area. And this is all occurring in a spatially recognized manner where literally every analyte, so as you notice, this has it's actually 1955.6976. These are very high-end mass spectrometers. And so being able to tell the difference between 1955.69 and 1955.89 is an important thing and easily done. And so again, we're generating huge amounts of data that really encompasses, literally thousands and thousands of molecules that are actually, we know they're sugars because we use an enzyme to release them over that tissue and in that cancer. And so I'll just show you an idea of what we did to start with. And so we use generally what we call training sets, discovery sets of tumor tissue, and then validation sets. And so this was a sample set that I believe this set came from a colleague at Pittsburgh, Paul Monga. And it was, included, a bunch of tumors that were primarily male, primarily driven, by cirrhosis primarily, but again, a significant portion didn't have cirrhosis at all, which is something we're seeing. And the tumors were, had a range of differentiation. Unfortunately, most of this is still, moderately, differentiated. And so this was used as our training cohort, where we then looked at the sugars that were present in the tumor area and in the adjacent, and in this workflow. And then I'm going to show you what are called UMAPs, where we break out all the sugars that we see in the tumor and the adjacent for patients and kind of get an idea of how easy the sugars differentiate between the tumor and the adjacent tissue, and then do that for all the patients. And the data itself is kind of interesting, and the UMAPs are confusing looking to begin with, but I'll explain them hopefully in a way that just makes sense. But please flag me if it's confusing for you. Um, and so that's how we're going to show it going on, the tumor, the adjacent that way, where the tumors will be, all the glycans found in the tumor on a spot by spot basis will be shown, and the adjacents will be shown as well. So in a start, just to kind of show you what we see generally, and is these are four different tumor tissues, A, B, F, and G, and I actually have a bunch more that I've cropped off here. Um, but basically what you see is, hey, here's this tumor in A, and the pathologist has annotated this tumor, and it has a sugar that has a molecular weight value, and I haven't shown the sugar here, but of say 2320 that's highly present in the tumor, but not in the adjacent. And there's other sugars there as well. And in this tumor down here, turns out it has a sugar that's different. It's actually at 1257, and that's primarily in the tumor. And then this tumor has the 1257 sugar, but it also has this 2393 tumor sugar. And then this tumor has a little bit of the 1257, but a lot of the 2393 sugar. So literally what I'm kind of showing you is that, hey, this tumor is kind of like this tumor, but not the same. And this tumor is kind of like this tumor, but not the same. And this tumor is kind of like that tumor, but not the same. And that's a key point and something that I want to highlight here. And so when we start looking at these UMAPs, what you end up getting is uh, in a UMAP here, I'm just showing four patients. And in the blue are the tumor glycans, and the orange or the adjacent glycans found in the adjacent areas. And we do this on a spot by spot basis. And so what you'll see is that the blues and the orange spread out in some cases like beautifully. Like here's all like the tumor and here's the adjacent. You kind of look at that and think, well, that's kind of easy. And you look at here and you think they're separated out here and they're separated out here. It looks like, yeah, it makes sense that no wonder and I said that, here CA19-9 is used as a biomarker. It's a sugar. And you could look at this and say, well, this looks pretty simple that they're differentiating themselves very, very nicely. So there must be no major issue here. These biomarkers, these glycans, these sugars on proteins make good biomarkers from, you can see that here. The problem is this, and this is what we've learned. So what this is, and this is the confusing picture, it looks like some abstract painting from Pollock from the 80s, but really what it is all the sugars that are seen in just the cancer tissue from the 86 patients. And what you see is this, is that there's a gamish of samples here. These patients here all have the same sugars, are kind of similar sugars. But there are a lot of patients that are all over the place. What it is it's heterogeneity. It's tumor heterogeneity. It's also glycan heterogeneity. So the my tumor may not be your tumor, and it may not be somebody else's tumor. There's so much heterogeneity in the tumor, and that heterogeneity in the tumor is now reflected in the actual um, glycan profile that you see within the tumor. That's why you may identify one sugar. So CA19-9, for example, I'm just using an example, is just one sugar. It would pick up maybe these guys in here in this mesh, but it would never pick up everybody else out here. You can't just look at one thing. You can't just look at two things. You can't just look at three things. You can't even look at 10 things to gain everybody with that has liver cancer. There's too much heterogeneity. So that's a fundamental problem. And that required us to come up with an entirely new platform to now allow us to look at literally everything all at once. And that's kind of what we've tried to do. And so looking at this, the key point that I would make here is there is significant heterogeneity in tumors with a glycan profile. And using just one sugar will never be enough. Looking at just 10 sugars will never be enough. We have to develop a way to look at all of this at once from patients in a time-dependent manner. So this slide, I will just tell you, took a student looking at 86 tumor samples. It took him, I think, probably a year and a half to develop. So the tissue analysis that we do is not suitable for real biomarker use. It's never going to be applicable to a patient in in a biomarker setting. It could be useful for other things, but we'll get to that in a second. And I will tell you, the one thing he did do, and so the student that did this is likes AI models, and I'll show you some of the stuff he's done. But he basically developed methods where he could really develop an algorithm that could then predict just looking at pathology and glycan analysis, um, identify tumors quite easily. And so he uses a machine learning based algorithm called XGBoost. He likes that. And so we trained it on a spot by spot basis of all the samples we looked at. And when you think about this, when you have a spot at 5 microns in a tumor, there are actually millions of actually tumor spots within that tumor tissue. And so when he did that, he developed an algorithm that used about 20 features, so 20 different glycans. And he developed a machine learning based algorithm. This is all AI now. And he could develop ROC curves that very accurately predicted the location of the tumor. And what he then did is he applied the same algorithm to the tumor tissue. And I don't want to, so unfortunately, he used the same color map. And so this is actually not some mass spec imaging. It's actually just a heat map generated by the application of an algorithm on top of the tumor tissue where we generated data from. And as you can see, where it highlights tumors being present, the pathologist could then go back and say, yes, tumors are present in those areas and not in other areas. And so we did this initially on the same set we used for discovery. And this was done on a pixel by pixel basis. And then he went back and used this on roughly 69 tissues from an external set. That these came from Texas, from UT Southwest. The first set, or discovery set, came from Pittsburgh, and this came from Texas. There are some differences, but literally, uh, uh, this again is not any sort of glycan imaging or any analysis. It's actually just a heat map showing the application of an algorithm on top of the tumor tissue, where red tells you the algorithm says that that is most likely tumor tissue. And as you can see, um, like this one in the middle, that big red spot is actually the tumor. In this thing, that red spot's the tumor. In this thing, the red spot's the tumor. In this thing, the red spot is the tumor. And so the algorithm predicts the tumors really, really well. In this case, for example, there were no tumors. So we actually got nothing there. This guy had no tumors, nothing there. Um, this one we're not sure about. We have to work on that. So it's not perfect yet, but it's gotten very, very good at identifying tumors just based upon an algorithm. Now, a pathologist can do this as well. And so what the true usefulness is, we'll see. And we're trying to see if this can correlate, for example, with outcome and other things potentially that may indicate, some degree of pathology. Um, but just the concept of being able to do digital pathology using AI, using these type of markers, is something that we're interested in pursuing. So the question then became, could we still see those those sugar changes that we saw in the tissue? Could we see them in matching serum from the same patients? And so we had developed another method that allowed us to look at the sugars in serum, and we've been doing this for many years. And so we had the tissue of the patients, that we knew the sugars that were there, and then we looked at the serum of those same patients. And to make a very long story short, we can. And so this method we've developed and published, but literally many of the same sugars that we saw in the tissue, we could see in the serum. So here's the serum level on the y-axis here and the tumor tissue on the x. And literally many of these same sugars that we saw in tumor, we saw in serum, literally almost at an R value of 1. I mean, it's literally, if it's high in the tumor, it's high in the serum. If it's low in the tissue, it's low in the serum. And so that was very comforting. And there were about initially about, oh, I don't know, about 14 or 15 sugars that we saw that correlated really nicely. We've looked at a whole bunch more, and there are lots. And so it varies from patient to patient, but there are lots of sugars that you see in tissue that are also found in the serum of patients with liver cancer. And that makes sense because the liver secretes many proteins into the blood, and many of the proteins that are secreted from the tumor also appear in the blood, and so we can see that. So that's great, but now what do we do? So how do we exploit this fact? So one of the things we did right away was to do some basically glycoproteomics, and I won't go into too much detail at this, but we have some workflows that we've developed that allow us to look at classes of proteins that that identify those proteins that contain those sugars that change. So we do this both in MUSC and then with a colleague at Mayo. Um, and we identified a whole bunch of proteins that are altered in the serum of patients with liver cancer with the same sugar that you see in the tumor tissue. And we have a list now of new proteins that, here the list is actually maybe 160 proteins long, and this I cut it off at about 78 just on here, but there's a whole list of proteins. So then this begs the question, how do we look at all of these things at once? So 20 years ago, what we would do is, and what we did is we'd pick one of these and look at them, or two of these and look at them. And that's a recipe for failure because what I showed you before was that there's too much heterogeneity within HCC, that one person's tumor is not another person's tumor. You may get 30% or 40%, but you're never going to get better than that. We have to develop a method that allows us to look at all these proteins at once. And that's kind of what we developed at MUSC. And so that's a method we developed called the glycotyper. And so our logic was simply this. And so um, when we use this fancy mass spectrometer to look at this tumor tissue, that machine, that instrument doesn't know what it's looking at. It just assigns to the slide an XY value to every spot. So if it fires a laser in the top left corner, that has an XY value. If it fires a laser in the bottom right corner, that has an XY coordinate. It has no clue what it's looking at. It just fires the laser at an XY coordinate. So we realized that, we could leverage this by, instead of putting in a tissue culture slide, developing this method to allow for the analysis of antibody arrays. So antibody arrays have been around for now 30 years probably, and all they are spotted antibodies that capture proteins of interest. And so our logic was, well, that is useful for us because we have literally, 150 odd proteins, 200 proteins we want to look at. And so if we could spot those proteins down, we could look at the proteins and then by, using our imaging methods that we developed, look at all the sugars attached to a given protein and therefore generate large amounts of data to make a better algorithm. And so we developed a method, and that's actually just showing what it looks like when the laser fires across the tissue. It generates what's called mass spectra. And it doesn't care what you look at, so it develops that. So we developed a method called the glycotyper. And so in this method, we create an antibody array. You spot the antibody across the slide to create the array, and this can be done manually by hand, or I'll show you where it's now done by a special printer. So that those antibodies are then added. So you then add serum, and those proteins, those antibodies then capture the proteins of interest that you're interested in looking at. So if you want if you want this protein or that protein, it can then capture them. We then add our PNGSF prime that then releases the sugar, and so that we know what sugar and what sugar biomarker is attached to what protein because it stays localized to that given area. And then we just code it and treat it like normal. And this is a new method that we developed. And so, and that method allows us to look at this. And so we then went back and then we decided to say, okay, let's use this method and to see if we can develop sort of a biomarker platform for this. And so we utilized a discovery set from UCSD. These are all blinded sets, and I'll tell you one of the problems you have with now machine learning and AI, they're too good in some ways. And so it'll haunt us. But we used a discovery set that came from UCSD, from Yuko Kono. And then we used an external validation set that came from UT Southwest. And there were about, say, 230 patients in the discovery set and about 160 in our external validation set. Um, you'll notice that there are some differences. In the discovery set, the cancers are actually larger than in the external set. If you notice in the discovery set, they're actually more viral in the discovery set and less so in the validation set. And that's something that's definitely going to come back to haunt us, and you'll see that in a second. And so here's what this data looks like. And so literally, I'm just showing you here. We spotted initially, I'm showing just 16 glycoproteins we looked at. And so like circle 1, 2, 3, 4, 5, 6, 7, 8, and down the line represent different antibodies that have been spotted onto basically a glass slide. It's a special slide. And then what is important is that when you then do the analysis, the signal stays where the antibody is. And so you can say that, oh, for example, in this protein, haptoglobin, with this sugar, what's the level in all the patients we looked at in the cirrhotics or cancers, and you can see there's a difference in the cancer. It's not huge, but it's there. For transferrin, this sugar goes down. For angiotensinogen, this goes up. For hemopexin, this goes up, whatever. And so you can look at these proteins on individual glycan, but you'll find that no one protein and no one sugar looks good as a biomarker. But that's important because, for example, in haptoglobin, this thing here, 2539, represents one sugar. There are actually about 50 sugars that are on haptoglobin. So we can look at all of them. There are on transferrin, not just one sugar, there are actually 50 to 60 sugars on transferrin, and so on. So each one of these proteins theoretically has anywhere from 50 to 60 sugars attached to it. And therefore, you start getting large data. So in this case, we have a list that's almost 1,700 things long. And those can all be put into combinations that can make a biomarker. And now that's not something that, again, you do normally. These are all machine learning based algorithms that are developed using high-end AI tools, and they put together the best combination and then they apply them. And so when you do that, what we ended up getting is, just checking my time, so a pretty good performance. Not perfect, and I don't understand why we're never perfect. It doesn't make sense to me, but we're never perfect. But you get a system where you get, literally 90 to 95% detection of all cancers with very few false positives. And that's true in both early cancers and all cancers and in cancers, and they completely outperform the current biomarker AFP, which we've often done. And in the external validation set, we're not as good. And the problem, I think, is simply this, is that what we learned is that these high-end machine learning algorithms are very good at defining a specific population. As I mentioned, did I show that here? So our big problem was simply this, is that our discovery set has all late cancers, a lot of late cancers, and our validation set has lots of early cancers. Our etiology is heavily viral in discovery and heavily alcohol and NAFL in our external validation. And so the algorithms are become very specific. And so we'd actually developed an algorithm many years ago, actually in Doylestown. It's called the Doylestown algorithm, and it's actually used by many people. And we developed that algorithm primarily using a set of virally induced samples, and it actually works very well for virally induced liver disease, but not for other forms of liver disease, which is not surprising. And so that's the one issue. So we're now working. We have a new grant. We're working with our longtime collaborator, Amit Singhal at UT Southwest, and Paul Monga now at Pitt, to get more samples and really refine these algorithms to make them better. One thing that we have done, though, is we tried to improve this technology. And so everything we've done in the lab with this method has been sort of a research grade. So literally a lot of it was being hand spotted. So we had spun out this technology to a spin-out company that we owned called Glycopath. And we got some grant funding. We then hired people to really develop it. These were mass spec people that came from BMS and other places. And they took what we were doing and they sort of supercharged it. So for example, when we were putting down, so by hand, we had to put down about 100 nanograms or 150 nanograms of protein or antibody, because that's all we could do because we have our hand pipetting. By doing a printer, these guys have gotten it down to 0.5, 0.15 nanograms. So literally, they've reduced it by, orders of magnitude, multiple orders of magnitude. And so, when we were putting down slides, we could only do a certain amount. They've now been able to put down, over 3,800 proteins on a slide to capture. So when we were only looking at, say, 100 proteins, they're now looking at 3,000 proteins at once. What they've also been able to do is this, because they've shrunk things down and amplified it. They've now been able to, look at combinations of proteins instead of just looking at 1,700 like we were doing at, they're looking at over 460,000 analytes at once. And so it's again an order of magnitude more. And they're now leveraging the power of AI to look at all these combinations. So again, these are now combinations that you can't even think about. So they're doing AI-based machine learning to actually make these combinations. This technology was recently purchased by Bruker Daltonics. So Bruker makes all the mass specs we look at, and they're actually developing this now as a platform technology to provide to other people as well to then validate. And we actually have two grants, one for hepatocellular carcinoma and one for cholangiocarcinoma, that's going to use this technology to sort of develop new biomarkers and then validate them with colleagues at Mayo as biomarkers for both hepatocellular carcinoma and hopefully cholangiocarcinoma. So with that, hopefully I haven't gone too long, and I try not to talk too long. I know I want to leave some time for questions. So the key takeaway slides were this, is that N-cyclic glycosylation, the sugars on proteins that I talked about, it's a molecular change that occurs in HCC, and it could also be observed in the serum. What I didn't really show you today is we know that all that variation we see in the sugars that are on the tumor tissue, it's directly related to the genetics of the tumor. And so I didn't go into too much detail about that, but we know that we've actually looked at that. The genetics, the underlying genetics of the tumor drive the metabolic shift that occurs in the tumor, and that's reflected in the sugars we see on the tumor, and we can do that. We've developed this new platform technology that uses antibody arrays that allows us to now look at thousands of proteins at once. So literally, we can now look at almost everything in the blood at once for the glycan. That whole assay takes 4 hours, and you can do literally, multiple at a time. So it's pretty neat, I think, what we've been able to do. And we believe this is going to be a new platform technology that allows us to kind of look for biomarkers in a similar way, because what we see in hepatocellular carcinoma and cholangiocarcinoma, we have colleagues that see in breast cancer, we have colleagues that see in pancreatic cancer, we have colleagues that see in lung cancer. So this is something that's not just it's not just linked to liver cancer, it's actually seen in many other cancers. And a fundamental reason, the reason is that it's linked with metabolism. And so as the metabolism of a cancer cell changes, the sugars that are on that cell and that are then coming from that cell change. And that's now something that's not our finding. It's been well known by many people, but it's the reason we see these things, and they make biomarkers out of it. With that, I'll thank a bunch of people. I think I've got most people here at the right place. I apologize if it's not. Some of this is actually very old. But I'd also like to thank, to a large extent, the patients that give themselves. So nothing that we'd have been able to do is possible without somebody who's given either blood or tissue for scientific discovery, scientific research, and really everything we do is indebted to them to get those samples to actually do that. So with that, I thank you for listening. Hopefully that made sense. I apologize if it didn't, but I'm happy to take questions and answer any questions that anybody has. And I thank you. So I'll stop sharing to answer questions, hopefully see people.
Great. Thank you so much, Dr. Mehta. We really appreciate this. Is such a rich presentation and really impressive and important implications. So thank you so much. We do have several questions that came in from the audience, several from Dr. Block. So I'll start with the first one. If you could please describe what is the basis for the 10 micron limit, and if you could improve resolution.
So the 10 micron limitation right now is the instrument. And so the manufacturer is improving the resolution, but they want to charge us lots of money to buy the new instrument. And so, but it's getting smaller and smaller. And so the new instruments are now at 5 microns. There are new ones coming out. They believe they can get the submicron. So they are being developed, and that hopefully will hit market and we can use those, but they are getting to that point.
Perfect.
Yep.
Thank you. The next comment was, I know you have thought about this, but would you comment and identify transferases that make the glycans? that may be enriched or unique in the cancers and can serve as either markers themselves or therapeutic targets?
Yeah, so that's again very good question. So one of the common transferases that we see that's upregulated in say 50% of cancers is what's known as fucosyl eight or FUT8. And that's something that we have, you know, been very interested in. We've actually made a knockout mouse that where hepatocytes have this gene deleted. And those animals that have FUT8 deleted don't develop liver cancer in one model. Um, and um, it appears that gene, oh, sorry, that gene, that's my timer to make sure I didn't go late. That gene is actually involved in all things in lipid metabolism. And so it's actually driving certain lipogenic pathways for the cancer to get it lipids to drive to use as an energy source. And so, um, and there are other genes that we do see as well that could be potentially both, I think, therapeutic targets, but also diagnostic targets. So yeah. So we would love for someone to make a target for that gene. Um, and the mice also, I will say, in a quite interesting manner, in a high-fat diet, don't put on weight, which is kind of interesting. Um, like the wild type mice. So, um,
Okay, perfect. We have someone who is wondering if you could please explain the details of the plot that showed the straight lines for the comparison between tissues and serum, and if those lines are just visualizations
they're
of the
actually
correlation coefficient.
so they are visualization, but the correlation coefficients are actually, and some of them are like 0.98, 0.97, 0.89. So they're actually incredibly strong. And so, so some of the glycans, yeah, they're like ridiculously correlative between the serum and the tissue, and um, that's to a certain extent almost in some ways expected because the liver is still pumping out lots of proteins into the serum. And so there is a normalization factor there too, I will say, and I forget what my statistician actually did. And that's done because the blood, when you look at it, does contain mostly material from non-tumor regions because we're looking at most of the patients we look at are small. They're small tumors. And so he does normalize it in some way, but there's a strong correlation there. Yep.
Okay, perfect. And then the last audience question that we have so far is, I think also from Dr. Block, it seems that many or most of the proteins with the HCC selective glycans are acute phase proteins. So are these coming from the tumor and not the surrounding tissue? It would seem so from the spatial work. If you could further explain that.
Yeah. So they look to be coming from the tumor itself. And so most of the sugars that we see um, are from the tumor itself. And the acute phase proteins are still made there in a good bit, in a good level. And we actually have, um, we're extending the proteomics in many ways. Um, and there are now lots and lots of proteins that are even not acute phase proteins that we're seeing that are actually being you know, all barely modified in the tumor tissue and are making their way out in both in the blood and actually, of all places, the bile. And we're back to the bile, but other places as well. So, yep.
Okay, perfect. And then let's see. I was also just curious how close this is to sort of widespread clinical use, and if there's also a cost, if you know anything about the cost saving
Yeah.
measures that this could have compared to like ultrasound or other more
Well,
typical.
so that's a good question. That's the harder question. I mean, the biggest limitation is right now, we use a very high-end mass spectrometer. And so they're common in mass spec facilities, but it's still $1 million. It's not something that's
Mhm.
going to be widely available. So what our industrial partner is trying to do is port it to a less expensive. So they actually have clinical mass spectrometers that are used in clinical settings, and they're actually porting it to that instrument. And literally push that as sort of a platform that people can use for this sort of analysis. And they're actually making it available as a research grade reagent right away. And so they're actually now commercializing it so that
Mhm.
people can use this for their own studies. And um, So there are people we know now at Mayo and at Pitt and um, Harvard and Austin, Texas, using this now for their own stuff. So hopefully they'll find something as well. And so as more people use it, hopefully it'll then be brought to the market. Um, but that's the thinking. So we're still a ways away to get it to be cost effective. Um, but
Sure.
but that's the plan. So
Very cool. Yeah, it's very, very exciting. We also had another question come in from Giovanni Viscona, who is asking if you see ways to expand the information captured by your method by including molecules other than glycoproteins, such as, for example, metabolites.
yep. So you can actually look at metabolite the spatial omics using metabolites is actually a little bit more challenging, but um, you can do it. And we've been doing a lot of that on using a different method for single cell analysis we developed, but you can do it. You can look at proteins. You can look at extracellular matrix. Like we have a colleague that works with us, Peggy Angel, who's a professor as well, who specifically looks at the extracellular matrix and uses enzymes that cleave the extracellular matrix. And actually, that's very predictive of tumor aggressiveness, and so in both breast cancer and liver cancer. So yes, you can look at many different things, whatever you really want to look at. And so we look at sugars as we do, but other people look at O-linked glycopeptides. Other people look at proteins, metabolites, neurotransmitters, drugs, everything. So yeah, these instruments are very amenable to many different technologies and many different metabolites. Sorry.
Wonderful. Well, thank you so much. This has been such a great and enlightening presentation and really, really exciting and cutting edge research that can have really tremendous implications for people with liver cancer. So we really, truly appreciate your insights. I see some folks in the chat also echo these sentiments as well. So, so thank you so much again. I know we're coming up to the top of the hour. So I just wanted to just wrap up by just again saying thank you so much to Dr. Mehta for sharing and to everyone today for participating in today's webinar. We hope that you found it to be informative and valuable to your work or life and can apply some of this information going forward or use this as a springboard. Again, a recording of this webinar will be shared out with everyone who registered to attend. And you can feel free to visit our Liver Cancer Connect website to learn more about our program and reach out to us for any additional information. And thank you so much again for joining today. And thank you, Dr. Mehta, for sharing. Truly appreciate it.
Not all. Thank you.
Thank
Take care.
you. Take care. Have a good one.